Good morning, good afternoon, and good evening to nearly 300 people across the world from 37 different countries located on 6 continents. I'm Jennifer Smilowitz. I'm the Director of Scientific and Strategic Development for the International Milk Genomics Consortium, otherwise known as the IMGC. And I'm excited to bring to you our annual spring Roll Up One Sleeves annual workshop. This workshop is hosted live, but it is also recorded, and the recording will be available to you around June 1st. We'll let you know when it's available. You can access it on our site. Before we begin the workshop, I'd like to share a little information about the consortium with you. So as you may know, we are a scientific society created in 2004 by Professor Sherman at UC Davis, along with some colleagues. And the discovery aimed to focus on was the sequencing the bovine lactation genome, which they accomplished in 2009. Today, the IMGC is made up of hundreds of scientists and innovators from across the world, from academia, industry, and other sectors, who work on the common goal to advance the field of lactation and milk science for human health. Now, we advance the field by leveraging 4 platforms. The first is our flagship conference that's hosted in the fall, and it features cutting-edge scientific research that accelerates scientific innovation. Our symposium attracts a diversity of disciplines and fields and fosters collaboration and increases the impact of publications in milk science. The second platform is our awards program, which highlights scientists from across various career timelines, from students to legacy pioneers in the field. And then this platform encourages ongoing engagement with our base. The third platform is our bi-monthly publication called Splash Milk Science Update, which translates the latest discoveries in milk science from primary scientific peer-reviewed literature and is written by professional science writers. We have published more than 450 articles on milk science that are searchable on our website. And finally, our fourth platform applies to today's event, and that's our annual Roll Up One Sleeves workshop. It's a practical, focused 3-hour webinar that addresses the translational issues specific to scaling up, analyzing, and navigating regulatory for milk components. And the goal is that participants come away with a better understanding of bringing discoveries to practice as concepts or products for human health. Now, based on popular demand by last year's attendee responses on the exit survey, this year's milk topic is extracellular vesicles and microRNAs in milk. To help us select next year's topic, please take 5 minutes of your time to complete the exit survey with the QR code here. Now, the workshop is hosted as a webinar with back-to-back talks by our 4 world experts in this field: Professor Marco Walben, Dr. Ray Newland, Dr. Spencer Marsh, and Dr. Stephen Hicks. They will all share with you their expertise and learning lessons on scaling up, analyzing, and navigating the health and regulatory aspects of milk extracellular vesicles and microRNAs. After Dr. Hicks' talk is over, we will have a 5-minute break, and then the panelists will join an open panel discussion for about an hour. Please submit your questions or comments anytime throughout the workshop in the Q&A box in Zoom, which will be included at the end of the webinar in our discussion panel. Without further ado, I'm honored to introduce to you our moderator of the webinar, Professor Nurit Argov-Argaman at the Hebrew University of Jerusalem in Israel, who has more than 15 years of research experience in lactation physiology with an emphasis on milk fat and membranes. Take it away, Nurit.
Thank you so much, Jennifer. Indeed, very excited to be here and moderate this workshop on this rapidly expanding field of research in the academia, as well in the industry, in the R&D units of many companies around the world, exosomes. So as a first speaker, I'm excited to welcome Professor Marco Walben, Professor of Veterinary Medicine, the Department of Biomolecular Health Sciences at Utrecht University. Marco, the floor is yours.
My name is Marka Wouben, and I am very honored to be the first speaker on this workshop. And before I go into the real challenges that has been asked by the organizers, I would like to give a short introduction on extracellular vesicles. And so we are very interested in deciphering the physiological roles of milk extracellular vesicles. And as you all working on milk, you know that what you eat in the first day makes really a difference for the rest of your life. And breast milk is nature's first functional food. And we all know that milk contains many factors that are involved in maintenance of immune homeostasis and also prevention of excessive inflammation and disintegration of the barrier. Before I start, I want to disclose that during many of our collaborations, so you can see here a lot of people with whom we have been collaborating on the milk research that we are doing for more than 10 years. We also collaborated with Friesland Campina and also with Danone Nutrition Research, and they both provided finance for doing some of our research. So within my own lab, several postdocs, PhD students and technicians have been doing a lot of work, and I don't want to go into detail on all the scientific details, but I really want to focus on the challenges. Before doing that, a short introduction. What are extracellular vesicles? EVs are multi-component vesicles that are released by all kinds of cells in the environment. And in general, you can say that these vesicles play a role in homeostasis, either in cellular homeostasis, but also in the homeostasis maintained by communication between different cells, so intercellular communication. The extracellular vesicles, they are described by the International Society of Extracellular Vesicles as vesicles enclosed by a lipid bilayer. And you can see that in the lipid bilayer, protein components are present there. The family of the tetraspanins is very common, be there as membrane spanning proteins, but you also have the intraluminal proteins and proteins associated to the lipid bilayer. What is important is that not only proteins are there, you can also see selective incorporation of nucleic acids and also metabolites, and that these vesicles are not static. They are cell type and environment specific components are in there, and it is highly regulated. So since it is tightly regulated, the whole cargo of these vesicles, many different EV subsets are released by cells, and that makes that when you study extracellular vesicles, you study a very heterogeneous population. What is important to remember is that extracellular vesicle-mediated communication is conserved in evolution. It is not something from just animals, but you can also see that bacteria, they can communicate via vesicles, also parasites, also in plants, and importantly, it is not only within an organism, but also between organisms that vesicles can play a role in translating and bringing over certain biological messages. And that was the reason why my group was very interested in to see like whether vesicles in milk could be a way how mother shows the newborn to cope with the new environment. So an extension of mammals that after you have given birth, you can still communicate with the offspring. If you want to study these extracellular vesicles, then of course you need to study milk. And yeah, breast milk is a very complex, and I call it a living fluid, because in milk you also have a lot of cells present. So cells from the mother, like also neutrophils, lymphocytes, macrophages, but also microbiota are present in milk. And all these cells can give rise also to extracellular vesicles. Beside that, you have these specific milk colloidal structures. like the milk fat globules, the casein micelles, and also the protein aggregates. So it is a really complex fluid to study. And what is also an important point to make here is that most of the time people look like milk, from the nutritional value, but all these colloidal structures, they have much more than just nutritional value. It is looking beyond nutritional value because these factors, they are involved in the maintenance of immune homeostasis and prevention of inflammation and disintegration of the barrier. So the first challenge that I defined was how to define a real EV-mediated effect in this plethora of all these colloidal structures, because you have these casein micelles, you have the milk fat globules, and besides that, you have the extracellular vesicles. But they have some partially overlapping characteristics, like their size can be overlapping, also the density can be overlapping. So when can we say that an effect is really an EV-mediated effect? I think that was the first challenge. And when we started to study milk, we first started trying to find a solution by setting up specific protocols to separate milk EVs from other milk components. That has been published already in 2014, a very nice protocol that we developed, and that was primarily also based on a kind of trial and error by using the, at that time, most common techniques for EV isolation and running into problems when working with milk. So we came up with a differential centrifugation protocol, and we did not pellet the milk because the pellets were really insoluble, but we converted the protocol by bringing the 10,000 G milk supernatant on top of the gradient. And you can see the first gradient described, shown here, is that by bringing the 10,000 G supernatant on top of the gradient, you could perform a density gradient and then collect the density fractions. What was equally important was thinking on a good procedure control, because when you look to an effect of some of these density fractions and you see a functional effect, how do you know it is a real EV-mediated effect? And for that reason, we always perform a procedural control. And the procedure control is shown here with the second gradient, in which we used the supernatant of the gradient after we have spun out the vesicles and followed a second time exactly the same procedure. By doing so, you make a real procedure control with having all the steps of your isolation protocol in there, and also possible other milk components that can still contribute to the effect that you are looking at. So for us, it was not only making the protocol of the isolation of the vesicles, but also thinking of the proper procedural control. And then the other challenge was, like, which concentration do you use in vitro assay if you want to study these milk vesicles? So which concentration are you going to use? After a lot of discussions, we said, like, okay, we do not know how much vesicles we need to add, for example, to a cell to see a certain effect. But by taking exactly the same volume of milk and having the vesicles collected in that particular volume after all the isolation procedures, we made an EV concentration that was similar to the concentration of EVs in milk, and we use that in the in vitro assays. So we use a concentration that resembles a concentration in milk. And of course, you always need to do dilutions to see whether effects are titratable. But this was a nice solution. When you perform then these assays, so you isolate these vesicles with these nice protocols, which take very long. It are lengthy protocols, but then in the end, you can separate vesicles from other colloidal structures quite well, not 100%, that is also clear, that you always will find back some case sign, lactoferrin, etc. But we had a very reproducible method to isolate these vesicles. If you then looked with cryo-EM, you still see that, of course, these vesicles are very heterogeneous. You never have exactly the same vesicles, but that is an intrinsic feature of working with extracellular vesicles. So the conclusion of challenge 1 is that the isolation of milk EVs is challenging and requires combined techniques to separate EVs from other colloidal milk components. Milk EVs are heterogeneous. You need to deal with that, but that is also probably why they are so effective, that you need this heterogeneity. Another point I do not touch upon, but of course it's clear to people working with milk, there are differences in milk composition between different species. And for each species, you need to check whether you need to have adaptations of these EV isolation protocols. So having defined such a protocol, then of course the next point was how to decipher the milk EV cargo function relationship. As I mentioned, EVs, they have a very rich cargo. All kinds of different biological molecules are in there, proteins, nucleic acids, metabolites, but which cargo is connected to which readout? I think that is one of the biggest questions. We are still working in the EV field and not only in the milk EV field. In 2021, we published a paper in JEF, and I picked some of the results out of that to really illustrate how we try to tackle this challenge. And what we found and published in this paper was that human milk EVs can enhance epithelial barrier function and dampen immune responses by modulating the epithelial cell migration and inhibiting endosomal toll-like receptor activation, and also inhibited CD4 T cell proliferation activation. And I want to go a bit further on this CD4 T cell activation to show how we try to connect cargo with function. Based on the findings we had, we postulated there that maternal EVs delivered via milk can create kind of window for regulated development of the GI tract and the immune system. Before I go into the connection between cargo and function, first, what we found with CD4 T cell activation, and this was a very simple assay, we just isolated T cells from peripheral blood. We stimulated them with CD28 and CD3 just to activate CD4 positive T cells, and we analyzed proliferation, activation markers, and cytokine production. On the right panel, you can see in the bar graph here that by the addition of extracellular vesicles, there is a strong reduction of the cell trace violet low cells, meaning that there was a very strong blocking of the induction of proliferation by the stimulus, by CD3, CD28. What we also show there, and here you can see that we always compare EVs with the EV depleted control. So that is the procedure control I explained before, that in all cases, the EVs, they lowered cytokine release of the cytokines shown here. When we, after exposure of these T cells with milk EVs, recover these T cells in vitro, and then stimulate them again with dendritic cells, then they had a normal response. So our conclusion was that milk EV transiently inhibited T cell activation, and that there was no evidence for the induction of regulatory T cells or tolerance. So it was a transient effect as long as the EVs, milk EVs were present. The next point is that we wanted to connect this then with the proteome of the vesicles. Martijn van Herwijnen from my group, in the meantime, had performed an extensive proteomics analysis of human milk EV vesicles and defined that these vesicles have a novel functional proteome distinct from other milk EV components. And by checking what kind of functions are related to these milk EV proteomes, you see a lot of cell-cell communication, signal transduction, etc. So the next step, what we did was to check which proteins were present in all of these milk EV donors, and then with enrichment, network, and functional annotation analysis, we wanted to connect the milk EV proteome with the functional outcome of the T-cell inhibition. So the question is, which milk EV proteins could be involved in modulation of T-cells? And when you draw this whole network, this integrated proteome and functional analysis, we could define some hotspots of regulation indicated here in red. And in blue, you can see all the milk EV proteins. This is a very busy slide. So if you make a schematic of that, it looks much easier. It looks like this. You can see that around certain hotspots in the signal transduction pathways, a lot of these milk EV proteins can interfere, can modulate. So that brought us to the conclusion that bioactive EV cargo hit hotspots in regulatory circuits at multiple levels. So our working hypothesis is now that the interplay between bioactive EV cargo at the target cell level determines the net effect of the EV-mediated modulations. So not looking for one protein, it is looking like which EV components can really react with the same hotspot. So this is then in the analysis function in relation to the proteome of the extracellular vesicles. And in the next couple of slides, I would like to show you also how we integrate the microRNAs in that. And that was when we looked to T-cell modulation by milk EVs derived from allergic or non-allergic mothers. In this work, this is not yet published, but we compared non-allergic milk donors with allergic milk donors. And we looked to the capacity to inhibit T-cell activation. What we found is that both allergic and non-allergic mother-derived milk vesicles could inhibit the proliferation, but you can also see that the allergic mothers, the EVs from allergic mothers, were less potent of inhibiting this proliferation, as can also be seen by the upregulation, less strong upregulation of the CD25. This is also seen in the cytokine profiles. We see both allergic and non-allergic, they downregulated certain cytokines. But for example, IL-6 and TNF-alpha, we could see a clear difference between non-allergic and allergic mothers. And the same was seen for transcription factors, for a cell cycle protein, messenger RNA for CDK4, messenger RNA for TGF-beta and IL-13, and also for these surface markers, CD40LG and the TNF SF14. So in general, we can say that there is a reduced inhibition of T-cell activation in the presence of milk EVs from allergic models. And then the next step was, of course, how can we connect that to the microRNAs and the proteins? So we look to differentially express microRNAs, differentially express proteins. And an important point is that we find some differentially expressed microRNAs and proteins, but that none of these microRNAs or proteins meet a significant threshold after multiple testing corrections. So the question is always, is that the end of the story? We think not, because as I mentioned before, the interplay of available bioactive EV cargo and ongoing molecular processes determines the net effect of T-cell modulation. So they can act in concert. And that was what the next step was to see whether we can integrate then the targets of the proteins and the targets, validate the targets of the microRNAs, whether they can hit at the same points in the cell cycles. And by doing these targets, we look explicitly to whether the targets were available, were present in T cells. The next step was the gene ontology analysis of T cell processes linked to the targets. And with that, you can plot up a whole scheme here where you can see these differentially expressed proteins and microRNAs acting. In general, we can see that T cell modulation is most pronounced by EVs from non-allergic milk donors. But then, of course, now the next step is to understand how that acts at the target cell modulation. So at the target cell level, EVs act in concert. EV cargos, they act in concert. So another point I did not address here is that it is also important to check whether EV cargo is available at the proper site and can contribute. So when you identify a certain target, but the cargo is not present at the proper place in the cell, then it cannot exert its action. So also that should be integrated in the final integration between cargo and functional modulation. And depending on the EV cargo types, some cargos can have direct and others have indirect effects. So there are also temporal differences. So the context and the activation status of the target cell are also important to understand, because that marks the signal transduction routes that are on in the target cell at a certain time that the milk EV cargo comes in. So that makes it really complicated to connect cargo, which cargo was inducing the final modulation. So what is needed now in the next step is that we need to know much more from single EV analysis and subsets identification to understand the heterogeneity and to get numbers on EV and cargo. But you can not just do this by hand. I think it is very important that in the future we are working much more with supervised machine learning and also deep learning, following a holistic approach to understand these cargo function relationships. So to reach the next level, I think team science is really essential, and that we need to team up with biology, physics, chemistry, and computational biology. And for working on these EVs, it is thinking big while working small. Now, this brings me to the last challenge. This is towards biomedical application. And knowing that these vesicles are so potent, there is a lot of interest to see whether they maybe can be used in baby formula, milk replacers, etc., to have good guidelines for nutrition, also for sustainable livestock, and also to see whether there are therapeutic applications for these vesicles. By going into that direction, it is there is an interesting point, is that from when you think about it, in general, we would like to say that the purer is the better. And this is an example of bovine milk, in which we try to make very pure vesicles by paying much more attention to get rid of the case-high mice cells. And you can do that with acidification, EDTA, or citrate. So we did all these protocols next to each other, and then we looked to purity. And the acidification resulted in a quite pure sample. But if you, for example, look then to function, the acidified EV samples, after they were neutralized again, they could not inhibit T cell proliferation anymore. So you have a pure sample, which is very useful for a lot of characterization, but it does not have the functional effect anymore. Then there is the other point, the safer the better. with a question mark. Here I show an example of in which we look to raw bovine milk and compare that with pasteurized, homogenized, and the ultra heat treated milk. To say like, okay, you want to have a safe starting fluid. By just doing this pasteurization, it did not look so bad when we look to the number of vesicles. But when we look to the microRNA, to the RNA concentration, we saw a strong drop already in the RNA concentration in vesicles derived from pasteurized compared to the raw milk. And for some protein markers, like for example, for CD9, there was not such a big difference, but with CD63, we saw a big change already in the population. So that could indicate that certain subsets of vesicles are more vulnerable than other subsets and that you lost some subsets. When we look to the integrity of the vesicles with cryo-EM, it was clear that the raw milk vesicles look really nice, but pasteurized vesicles already show some damage. So industrial processing of milk can influence EV integrity. And the conclusion from this is that I think milk deserves a real holistic approach. Raw milk is not the fluid that we buy in the supermarket. And processing habits are not primarily designed to maintain the bioactivity of the components in milk, and that there is a need for new technology in which there is more emphasis on the maintenance of these nature-assembled structure for optimal and specific delivery of bioactive components. Of course, without jeopardizing safety. So this is a very important point. So my conclusion and future direction, so this is my last slide. I think EVs are evolutionarily conserved and an integral part of biological regulatory circuits. But EVs are also vulnerable functional food components that can modulate cells in the gastrointestinal tract, including immune cells. And that it is important to isolate milk EVs by using really tailored protocols if you really want to pinpoint an EV-mediated effect. The cross-species mode of action opens a lot of possibilities for biomedical applications, and I think this is a bright future for the milk EVs. But a lot of things need to be done and standardized, and this is where I would like to draw some attention to the International Society for Extracellular Vesicles, in which there is a milk EV task force really focusing on all these needs for milk EVs to make more standardized protocols and guidelines for people working with milk EVs. The other task force is the Regulatory Affairs Task Force, in which there is a main task to see how we can bring these milk vesicles and other type of vesicles towards the clinics, towards therapeutics, etc. So there's quite some work to do, but I think it's a bright future for it. And this is the end of my talk. I thank you.
Thank you, Marka, on a fascinating talk, which includes also some review on extracellular vesicles in milk in general. And thank you all of our audience for the questions that you were posting. And don't forget to submit more of your questions on our Q&A box at the bottom panel on your screen. We will address them to our speakers during the Q&A session at the end of the workshop. So I'm happy to invite our next speaker, which is Rink Newland, Research Associate from Amsterdam University Medical Centers. Rink, the floor is yours.
Sorry for the slight delay. I'm Rink Newland. I have no relevant conflicts of interest. I'm from the Amsterdam University Medical Center, where we have a group, we call ourselves the Amsterdam Vesicle Center, where we work on isolation and detection, functional characterization, and standardization of EV research. In contrast to Marka, I'm not a milk expert, but I hope that the findings I will show you today will trigger your interest. So I have 3 questions I would like to address today. The first one is, are there any functional differences of milk vesicles between mammal species? And if you look at one mammal species, are there functional differences in body fluid-derived vesicles, so in one species? And is there any relevance for human health application? Now, I know much more about blood than about milk, I have to admit. And my favorite model is a bleeding finger. When you have a bleeding wound, that bleeding will stop when you don't have any deficiencies. And what happens is the following. So you have a damaged vessel wall. The platelets shown in the middle here adhere to the damaged vessel wall. The platelets make a clump, and you can compare this to bricks that are stapled one on each other. But then if you do that, this is a very loose construction. And then finally, this platelet plug stops the wound and is strengthened by fibrin strands. And these fibrin strands are insoluble protein polymers. You can compare it to mortar, and it glues the stones, the bricks, the platelets together. And it's about this process of fibrin formation I would like to talk and to explain to you a little bit. Now, if you look at coagulation or clotting, there are several requirements. And all these requirements for fibrin formation are present within your blood. So I'm talking about calcium ions, many coagulation factors, fibrinogen, that's the final substrate of the coagulation cascade, and the membrane surface. And for those of you who are not familiar with coagulation or blood clotting, if you prepare the blood fluid in the presence of an anticoagulant, the plasma, the blood fluid, is a clear yellow fluid. And then when the plasma clots, and it can really happen in a few seconds, the plasma becomes turbid, it's not a fluid anymore, and it's gel. And in this, here you have soluble proteins, and here you have the insoluble fibrin form. And this is really a very dramatic process. Now, if you look at coagulation components that are present within your blood, you have many, many different coagulation factors. And you do not have to learn this by heart. It's just good to realize that everything you see now is present within your blood. There's one component, however, that is missing, and that is a protein that is present only outside the blood vessels, and that protein is called tissue factor. And tissue factor is an interesting protein because what it does is it binds coagulation factor 7, so you have an extravascular protein that becomes exposed to the blood. It binds a coagulation factor, factor 7, that's activated to 7A. Then you activate 10A, thrombin is formed, and finally fibrin is formed. So the gel, the strands. Now, tissue factor is a transmembrane protein, but it's, as I said, it's an extravascular protein, and it is a transmembrane protein, so it's really membrane associated. It's a receptor for coagulation factor 7, and as I just showed you, coagulation factor 7 is an intravascular protein, so it's present within the blood. Tissue factor 7A complex triggers blood clotting under normal physiological conditions, and it requires phosphatidylserine for its activity, and that's a phospholipid that is usually present close by tissue factor in the membrane. Now, already since the early 1980s, we know that tissue factor is associated with extracellular vesicles. This is an example from pathology. I think this is the very first paper where tissue factor was not even described, but in a science paper it was shown that various types of tumors, both in vitro and ex vivo, release vesicles exposing a coagulant activity, and we now know that this activity is called tissue factor. This is an example from our own research. as I told you, tissue factor is an extravascular protein, but tissue factor can also be present in normal human body fluids under physiological conditions. And what we showed in the blood paper in 2011 is that if you have normal human saliva, we think that you expose the blood to saliva, so you lick your wound because the saliva contains large numbers of tissue factor exposing proteins. So what you're doing is you bring the initiator of coagulation together with the blood, and then the blood clotting goes faster. This is a very important mechanism, not only to prevent blood loss, but also to reduce the risk of infection. And you can do all sorts of simple experiments. You can just take normal human saliva, you put it in a centrifuge, and for example, in the supernatant, there's no tissue factor detectable, but in the pellets where the vesicles are present, we do see tissue factor. You can also do electron microscopy, and here you see tissue factor staining by transmission electron microscopy. So what this shows is that tissue factor, the protein that triggers blood clotting, can be present in normal human saliva, and it's very, very potent. And just to show you how potent this is, so here we are monitoring the reaction of plasma in a soluble to an insoluble form. So here's fibrin formed. And when you look, here is the optical density at 4 or 5 nanometer. This is just plasma that you pipette in an ELISA well. Here you add calcium ions. And what you see is that the plasma does not clot. So the plasma stays a fluid for 1 hour. If you add a few microliters of saliva or vesicles from saliva, you see that the optical density increases, and this reflects the conversion of the clear solution to the turbid solution here. So really fibrin is formed. So you see this is a very fast and very active process. Now we come finally to milk. Already in 1934, there was an observation by Alphonse Solé who showed that mother milk is very effective in stopping bleeding. And they did their experiments because they were faced with boys with hemophilia. They have a bleeding disorder. They are missing factor 8 or factor 9. They didn't know what to do with external bleeding wounds. And they applied gauzes soaked in various fluids. And one of these fluids was mother milk, and it was very active. And in fact, Edward Glansman in 1935, he is the father of the platelet research, now one of the fundamentals of hemostasis. He wrote, if you translate it from German, no other animal milk has such blessed hemostatic properties as human milk, as first shown and described by the Viennese pediatrician Alphonse Solé in 1934. Sorry. Put a tamponade soaked with human milk and press it on the bleeding spot. Now, another Austrian hematologist on the left, Johannes Stawe, came a few years ago to me, and he was asking me whether we could do experiments similar as we have done with saliva to see whether also human milk contains vesicles that expose coagulant tissue factor. And it's interesting because he visited the second wife of Alphonse Solé, who is sitting here on the right. And we published a paper a few years ago showing that human milk triggers coagulation by exposing tissue factor, exposing vesicles. So exactly similar to saliva, also human milk has these vesicles. And I should say, I don't show it now to you, but also if you collect tears or if you collect sweat or even urine, you will find exactly the same. And what we showed is this is a very simple experiment. On the y-axis, you see the clotting time. So if the bar here is 3,600 seconds, so 1 hour. So this is when you add buffer to plasma. The plasma does not clot for 1 hour and the bar stays high. This is the lowest concentration of milk. And then, sorry, losing my mouse. So the lowest concentration of milk, etc., increasing concentration of milk. And what you can see is that the clotting time decreases when you add more milk. In fact, Johannes calculated that you can dilute human milk about 4,000-fold, and you can still measure the clotting activity of human plasma. So it's a very potent activity. In this paper, and I will only show a few of the experiments we did, but one of the things we did, we fractionated human milk in 26 fractions. So these are the fractions to 26. These are fractions that we obtained by separating milk on size exclusion chromatography. This is a chromatography method that has a cutoff of about 70 nanometer, and in fraction 7, 8, 9 or 8, 9, 10, you see that we have, this is plotted for tissue factor. You see a tissue factor antigen here, these three fractions. It's not detectable in any of the other fractions. And these are the fractions that typically the extracellular vesicles are present. And the question, of course, is, if this is a tissue factor that's triggering clotting, is the coagulant activity really also in these fractions? So for each fraction, we looked at the ability to trigger the clotting of plasma. As you can see here, in the absence of any antibody, this is clearly limited to the fractions where we see the tissue factor antigen as well. So there's also the activity. And after pre-incubation with an antibody against tissue factor, you see that the clotting activity is completely abolished. So yes, there is tissue factor in human milk. It's associated with particles, very likely vesicles, and we can completely block this response. And what we think is that human milk, like saliva, contains vesicles exposing tissue factor. They bind factor 7 in blood. Factor 10 is activated thrombin, etc., and you get fibrin formed. Now, there's one thing I would like to add. More recently, we have shown that in reality, it's even more advanced. And we show that the coagulation factor 7, which we always thought is present only in the blood, is actually already present on the vesicles in milk. So if you isolate vesicles from human milk or saliva or urine, these vesicles not only expose tissue factor, but also already factor 7. And you can very easily demonstrate this by adding these vesicles to factor 7 deficient plasma, and then the plasma just clots very efficiently. So this was a surprise for us. So, so what we actually showed is that the vesicles in normal human breast milk have not only tissue factor, but they also have the tissue factor 7A complex. Hematologists call this the extrinsic tenase complex, and it's extremely potent in triggering activating factor 10A, thrombin, and fibrin. And in fact, this may be the reason that blood is such an extremely potent fluid to stop bleeding. Now, there was another interesting observation in 1934 or 35, and that was that bovine milk is very different and it does not stop bleeding. So, whereas human milk stops bleeding, bovine milk did not. In fact, they also looked at milk from guinea pigs and some other animals, and they all did not stop bleeding. Now, I think there are two likely explanations. And we thought in Amsterdam that the first explanation is the most likely, and that's the explanation that bovine tissue factor simply does not bind to human factor 7. So there's no cross-reactivity between species. And of course, there was a second explanation which we deemed very unlikely, and that is that bovine milk does not contain any tissue factor. Of course, there will be vesicles, but it might lack tissue factor activity. Well, we collected also bovine milk, and to our surprise, well, first of all, we did not see any tissue factor antigen on the Western blot. But again, keep in mind, we have antibody against human tissue factor, so perhaps it just does not cross-react, so this is a bit tricky. But the proof of the pudding is here. These are 3 bovine plasma samples where we added just buffer, or we added bovine milk, and you see there's absolutely no effect. The clotting time is really unaffected. And if you add human milk, so containing tissue factor, human tissue factor, you see a shortening of the clotting time. So it really seems that bovine milk has no tissue factor activity. Then, if you can still remember that I showed you the data from saliva, so licking a wound, cows are licking the wound. So we also collected bovine saliva. That's not so easy, I can tell you. They're the only green saliva samples in the fridge in the Amsterdam UMC, I guess. But if you collect bovine saliva and you add it to bovine plasma and compare to buffer, you can immediately see that there's a strong clotting activity present in bovine saliva, just as there is in human saliva. So if you compare these fluids, human and bovine, under physiological conditions, you see that in the human system, in humans, both milk and saliva have vesicles exposing highly coagulant tissue factor. Actually, they expose the extrinsic tenase complex of tissue factor and factor 7A. When you look at cows in milk, we cannot detect it. I think it's really not there. Again, these are normal, regular cows. I have no clue about colostrum, wild cows, etc. But if you take saliva, it is clearly there. So I think I wouldn't be surprised that also these other body fluids of cows have vesicles exposing coagulant tissue factor. So coming back to the challenges and the questions, are there any functional differences of milk EVs between mammals? I think the answer is yes. Whereas human milk is extremely coagulant, bovine milk lacks this activity completely. Are there functional differences in body fluids, body fluid derived EVs in one mammal species? Yes. If you look at let's say the saliva and the milk of cows, there's for sure a difference when you look at the tissue factor activity. And is there any relevance for human health applications future? I have 3 brief examples if I do have time left. One is screening of donor human milk pasteurization methods, use of human milk tissue factor activity as reference material, and the role of vesicles exposing the extrinsic tenase complex in intestinal epithelial barrier function. Now, screening of donor milk pasteurization methods study actually very simple. So what had been done already is samples, milk samples from 30 women had been collected in the AMC. And if we add these milk fractions to human plasma, so what you see here is again, you see on the y-axis the plasma clotting time. So this is again 1 hour. And what you see is that in these, for all these 30 samples, we also added buffer. So this is 30 times buffer. And what you see is that there's absolutely no clotting. So this is plasma with buffer, it doesn't clot. If you add, I think it was 5% milk from these 30 donors, you can see that the clotting time is nearly at baseline. It almost immediately clots. So there's a huge difference between the negative and the positive control. Now you see 3 methods, Holder pasteurization. This is a method involving pasteurization heating procedure, and this one as well. In all these 3 examples, you see that the tissue factor activity deteriorates. I think that's exactly what Marcus showed, that pasteurization may have detrimental effects. I don't know whether it's the vesicles or the tissue factor, but clearly something's going on. In contrast, if you look at high pressure pasteurization or you look at UVC irradiation, you see that these 2 methods are much more mild for the tissue factor activity than the methods where the vesicles are being heated. So it seems we can use the tissue factor activity in human milk as a readout for the effects of pasteurization. Coincidentally, very recently, I told you I'm much more involved in blood research. There's a lot of interest in measuring tissue factor activity in human blood and in human plasma. And because the tissue factor activity in human milk is extremely high, we as a core lab prepared a huge number of plasma samples containing a low and a high tissue factor activity. And these were all purified milk EVs. And I think this has been measured now in 30 or 40 different labs, and this paper has been recently submitted. So it's probably not the first thing you would think of, but it's really, even compared to saliva, the activity is amazing. And finally, and this comes also a bit back to what Marcus showed, we are having in AMC a model where fetal cells from the gut make a sort of organoids in a transfilter model. These organoids consist of multiple cell types, and you can measure tear and that sort of things, and you can look with a confocal microscope at the presence of cell types and how intact they are or not. And we are trying to answer two questions: do milk vesicles affect epithelial barrier function? And do milk EVs protect against inflammation-induced damage? And the first results look pretty promising. So these are the people who've done the work. On the left is Johannes Staler, Austrian hematologist, Ji Hao, technician in my lab. This is Jung Hu, a PhD student. René Bergmans, who did all the saliva work in the past. And this is René Scheurs, who did the experiments with the how do you say that, the fetal organoids. That's it. Thank you.
Thank you very much, Rink, for a fascinating talk and for contributing to our understanding on the field of EV from different biofluids like saliva and blood. And I find it especially interesting the fact that breast milk can act as a coagulant. And the fascinating fact is that women naturally apply breast milk on wounds of babies and newborns. And this is something that is done intuitively. And I think it's an amazing piece of information. So I just want to remind you to don't forget to submit your questions in the Q&A box, and we will address them to our speakers during the Q&A session at the end of the presentations. So our next speaker is is Spencer Marsh, Chief Scientist Officer from the Tiny Cargo Company. Spencer, take it away.
All right. Thank you very much. And thank you all for being here. I have to say, this is very easy for me going after those two talks. Typically, I have to give a ton of background on milk exosomes and the purpose and all of that. So thank you all. Today, we're going to talk about Tiny Cargo's milk exosome technology and the goals upon which we are trying to get to. So first, disclosures. I'm an employee at both Tiny Cargo as well as Virginia Tech. I did my postdoc at Virginia Tech prior to spinning off the Tiny Cargo company from that. And then from that, obviously, I'm a shareholder in Tiny Cargo as well. So Tiny Cargo's purpose is to utilize milk exosomes in both therapeutic and nutraceutical products. And most of the time, I really need to harp on why milk. There are a lot of groups that do exosome technology or extracellular vesicle technology, and they'll utilize mesenchymal stem cells and other cell populations in order to have more control. However, from one cow, it's estimated that you can get over 1E21, and that's more zeros than I can even really picture in my head, exosomes per year from that one cow. Indeed, as you look on the right, you can see a TEM image, and this is from a lab-produced sample from my time at Virginia Tech. And the EVs or exosomes are just packed in there like golf balls. And we can purify this from 1 liter of milk, and we end up with about 100 to 150 milliliters of concentrated exosomes at the density shown to the right. So you're saying, okay, there are that many in there, then what's the big deal? Well, I actually had some assistance, because the fact of the matter is that current processing techniques completely destroy the bioactivity of milk EVs. Now, if you look at this EM, you'll see that there are some vesicular structures. And this sample is actually from an industrial processor known as Fonterra. And we took Fonterra's dairy powders and processed them under our purification techniques. And what we found is that there are vesicular structures, but they do not maintain any of the bioactivity as far as wound healing, gastrointestinal regeneration, neurological development, et cetera, that milk exosomes have been shown fairly conclusively in the literature. So current processing techniques are going to destroy these. So what is your way around it, and what's the purpose? We are under an active MOU with Fonterra, as I mentioned, And we are currently in the middle of a contract with Food Pilot and Massey University in order to industrialize purification of milk exosomes from a starting solution of milk. And that is because Fonterra wants to utilize milk exosomes in products. And they want to see this as something that can be used in a bioactive ingredient or sold on its own. But the issue being that as of current, there aren't any milk exosomes in their products. We have this vesicles. These lipids are going to reassemble after being destroyed either in pasteurization, homogenization, concentration, or air drying, all of which are high temperature, high pressure treatments, which result in the complete loss of bioactivity. Now, what is this bioactivity? I've cited here, there's a group, I'm positive that they're in attendance right now, the Zemplenty group, who are really right now the gold standard of showing what milk exosomes do in bioactivity as far as in development or in regeneration. And in the study that I've shown here, there were two groups of mice pups, one that received very standard breast milk. The other received breast milk that exosomes were completely removed from. The group that received exosomes in their milk were able to solve maze puzzles 9 times faster than those that received milk without exosomes included. This is just one of a litany of examples in the literature that show the developmental and regenerative benefits of exosomes in both natural health and in the development of infants. Just as an aside, we have administered, and so we obviously use cow milk. We've administered cow milk exosomes in a number of mouse trials, and in every single one, the milk exosomes have a therapeutic benefit. We'll get into that near the end, but it's shown that this cross-species reactivity is extremely potent. So then there are a lot of exosomes there. They're destroyed during pasteurization. So what's your approach and how do you do this? Marka touched on this during her talk, and there are some methods currently that people use. And the primary problem behind this is that exosomes and casein micelles are attracted to one another and they adhere to one another at typical milk conditions. So what you're looking at with this EM, there are a number of vesicular structures that are exosomes, EVs, and most of them are bound within this net of casein. And at standard temperatures, casein is going to form and associate with itself and form these long filaments. And that is going to trap exosomes within. Now, I believe one or both of the previous speakers mentioned acidification of the milk samples in order to purify exosomes. And while this does work, the issue is that this net of casein, when you acidify, is going to precipitate. And when it precipitates, if the exosomes are still adhered or associated with that casein, you're going to be removing those as well. So in our experiences, when we have used acidification in the past, it results in much lower yields than other approaches that we're about to go over. So what exactly is the opportunity here? And that is exosomes are associated with casein via calcium-dependent bonding. And we use that theory to then enable the scaling in an industrial setting to greater than 1,000 liters per batch. How we do that exactly is using the other molecule that Marka mentioned, and that's EDTA. Now, this right here is a process that we have validated at 1 liter of input at the research scale, which is about 100 to 150 milliliters of output. And we are also in 2 weeks, I will be in New Zealand at the Food Pilot facilities working to industrialize this process right here. So what we do, it's very, very simple. You take fresh, unpasteurized milk, and I'd like to just mention Homestead Creamery in Wirtz, Virginia. They've been getting us raw milk for a long time now, and we appreciate them. We then defat this milk, very standard industry defatting. In the lab setting, we use about 5,000 RCF for 30 minutes at 4C. That's going to result in a thick fat pellet, or not pellet, on top, a fat layer, which you then just remove, take the liquid below, and treat with EDTA. The molarity of EDTA is very specific. 30 millimolar EDTA, the time and temperature are also very specific, 37 degrees for 60 minutes. After that 60-minute period, you can then take your solution and run it through a 0.45 and a 0.22, and then run it on what's known as a tangential flow filter. Tangential flow is the effective opposite of depth filtration, in which in depth filtration, you're just passing right through a filter And what you keep is the permeate, and you throw away the retinate because that's retained in the filter. In tangential flow, it's the opposite. You're passing the solution over a conical filter that removes things smaller as opposed to larger. Over that period of time, you'll then keep the retinate, not the permeate. The permeate will contain your EDTA, casein, micelles, whey protein, etc. And the retinate will contain your purified exosomes. At that point, it'll be about 10 to 15% of your starting volume. And then if you want a pharmaceutical grade version, you'll then need to run it through a Cephros column, 70 nanometer column, and you'll retain fractions 8 and 9. And at that point, you'll have exosomes at over 5E12 per milliliter at the density shown in the TEM on the right. So just to reiterate that, the steps of this, they're really, really simple. And this is what we've done in the research scale for a couple of years now. And we're now going to work with Food Pilot to scale up. And that's just standard defatting, treatment with EDTA, a couple of filters, tangential flow filters, and a Cephros column. But there's a problem. And the problem is that EDTA is naturally going to interfere with the casein products. And what I didn't understand when we began talking to Fonterra, as well as these other dairy companies, is that EDTA is not great. And casein is their golden product. Casein is where they get the vast majority of their financial windfalls. And they just can't have a process in which that casein, that really valuable byproduct, is going to be compromised in any way. So we were basically presented with, okay, you can come down and you can do your EDTA process, and that can be your pharmaceutical approach, and that can be, whatever you want to do. But if you want to move forward with us, you have to have something in which you're only using natural products to perform this purification. And I have to say, I'm sorry that I can't share with you exactly how we solved that problem just yet. We have IP that is pending, and we're going to validate this process at the Food Pilot facilities in just over 2 weeks. But we are using an isolation in which there are only natural products that are standard occurring in the milk to accomplish a complete dissociation between casein, micelles, and extracellular vesicles, resulting in a purity equivalent to our EDTA approach. And so that's all great. We have a process that we can now use, and this is validated at the research scale, and very soon it will be validated at industrial scales. And hopefully we'll have one that we can use in nutraceutical products with Fonterra. But this then arose another problem. If you're going to create a product for a international dairy conglomerate, it has to be a powder, and they can't just ship bottles of things all over the place. In fact, Fonterra pretty much dries all of their products, and then these are sold wholesale to other large dairy companies. But very similar to pasteurization and homogenization, the air concentration and drying steps, which are done at over 100 degrees C, and I think concentration is done at almost 200 degrees C, completely destroy the ability of our exosomes to maintain the microRNA contents that are responsible for these downstream effects. So how do we enable this? How can we dry these if the common approaches are completely insufficient in accomplishing this? Well, this problem was a bit easier to solve, actually. It turns out, and it's probably quite a few of you in the audience right now that are aware of this, but milk exosomes, shockingly, can be lyophilized very, very simply. If you add 50 millimolar Trehalose to those post-Cephros column exosomes that I just explained to you, you can then crank a lyophilizer to its highest level. You can take the condenser all the way down to minus 100. You can take the pressure all the way down to 10 millitorr. Which is a really aggressive lyophilization approach. And as long as you have about 50 millimolar Trehalose in there, we see complete bioactivity retention and about 95% particle count retention after months of sitting as a powder. And I like telling this story because it's a little ridiculous. The first time that we did our lyophilization approach, we had about 10 or 12 vials of different cryoprotectants and different things. And this was right before Thanksgiving. And so we lyophilized them, and they got put up on a shelf, and everyone forgot. So we forgot about this until, I want to say, mid-February. So it had been about 4 months. And one day I walk in and I just see the vials up on a shelf, and they're just sitting in the sun. And it hit me, oh my goodness, those have been sitting there and I completely forgot about them. And they've just been sitting in the sun, wasting away for months now. And it turns out, even being blasted with photons for months at a time, we saw complete retention of our bioactivity and complete retention, about 95% of our particles. So right here, you can see outputs from our EM in the top, and then from our nanoparticle tracking analysis on the bottom. The fresh is shown on the far left. You can see very nice EVs in there. Lyophilized, this is just in our buffer. We use a HEPES buffer. We're effectively totally destroyed. And with the addition of trehalose, we saw near complete retention. So at this point, you can purify exosomes from milk, huge volumes, huge yields, and simply lyophilize, store them, and save them as long as you really would like to do. But the nutraceutical side is fine, and this will enable use in the industry for nutraceutical products, but that's not our goal. When we started this research, it was not to create nutraceutical products. It was not to create dairy powders. This is a fortunate kind of byproduct of what our eventual goal is. And that's to use milk exosomes as a drug delivery platform. Now, I don't know how many of you were at the IMGC event in the fall, but much of my talk at that event was on the ability of milk EVs or milk exosomes to target injury. And most of this data was shown using either in vitro scratch assays, in which we scratch wounded and then applied fluorescent exosomes, or in vivo in a heart attack model. So we had a collaborator of ours induce what's known as an LAD, induced ischemic event. It's commonly known as a widowmaker heart attack. And then we administered fluorescent EVs, fluorescently tagged EVs. And what we found was 20 to 30 times increased expression in not only the injured organs, but the injured portion of those organs, which shows a very finely tuned ability of milk exosomes after oral administration to bypass through the gut into the bloodstream, avoid first pass metabolism, and target into injured organs. In this case, the heart, if not the most dynamic, one of the most dynamic environments that you can imagine inside the body. So what does that really mean? We have something that we can administer and it's an occurring drug delivery platform. That's great. It actually, and once again, here's data from the Zemplenni group, which is just a fantastic image showing the ability of these to bypass through the blood-brain barrier. So we've confirmed that orally administered milk EVs can bypass all these internal barriers and direct their administration to injured tissues, which is fantastic, but you need a cargo, and you need something that is actually a pharmaceutical grade cargo that can then be utilized in this situation. So how do you do that? How do you take a stable milk exosome and put what you want inside it while maintaining the ability of the EV to then be orally administered, bypass internal barriers, and be uptaken by cells of interest? Well, there are a bunch of different ways, and I would go out on a limb and guess that many of you here probably use one of these methods. Very typical approaches in the field: sonication, electroporation, extrusion, freeze thawing. And the idea behind all of these are effectively the same. That is, that you are going to porate the exosomal membrane to allow for the cargo to be placed within that membrane. And the idea is that these are temporary pores, because many of these approaches have been used in cells to load AAVs, et cetera. But the problem is an exosome and an EV isn't a cell. And I don't believe from our experiences, it does not appear that these temporarily porate the membrane. It appears that they permanently porate the membrane. So our approach really was to find a method that we could disrupt the membrane with no permanent damage. How can we create a temporary pore in an exosome which doesn't have a nucleus? It doesn't have the ability to fix itself. And this goes down a slightly complicated path. But what we fell on was what's known as a glass transition temperature. And transition temperatures are very widely used in material science engineering for metals and for liquids to show when they're going to change phase. And that's going to tell you about the phase boundary and the crystal structure of the metals. But in biology, what this tells you is that these temperatures are going to be different across various points of the membrane. certain parts of the exosomal membrane are very highly enriched in cholesterols. Others are very highly enriched in lipids, et cetera. And so this is going to create phase boundaries within the membrane. Now, through about a year of research, we identified, and we also found validation in the literature, that there are two glass transition temperatures for a milk EV membrane that are very easily accomplished. And that's 31.2 degrees Celsius and 42.0 degrees Celsius. And what we found was at these temperatures, we were able to dramatically increase the ability to bypass the membrane and stably load exosomes. Our final process now uses a kind of a moving between the two temperatures. We just kind of oscillate between them, which works very well. The second point of this is that milk exosomes contain a very large negative charge, which means that if we were to do our temperature variation, that oscillation, and put in something else negative, doesn't really matter how large the pore is, because it's just not going to be able to bypass through that membrane with any sort of pharmaceutical efficiency. Sure, you'd be able to get, 2, 3, 4, 5% maybe, but that's just not going to do it from a cost basis as far as developing a pharmaceutical product. There's a third piece here, and I know I told you this gets a little complicated. Milk exosomes contain what are called esterase enzymes, and these are endogenous within milk EVs. What you're seeing here is a dye called calcine AM. Calcine AM is an esterified dye that increases in fluorescence when it is deesterified. This is commonly used in live dead stains to illuminate living cells. What we've done is we've utilized this dye in our own assay to show whether or not REVs have patency. But a crucial component of this is that calcine must be deesterified. And that means that there are native esterase enzymes present within the exosome. Okay, so we got three major pieces here. We're utilizing glass transition temperatures to slightly porate. We can't be negatively charged, and there are esterase enzymes present. What that all adds up to is the ability for us to take a peptide, that's RPRP-DDLAI is the peptide that we're using, otherwise alpha-CT11, which is highly negatively charged on its own, and we cap those negative charges with methyl groups via ester bonds. Now that shielding of the negative charges creates a neutral peptide that, when incubated at a glass transition temperature, and you have this shifting and this small poration occurring, can simply bypass through the membrane because it's neutral. But the key to this is that the esterase enzymes present within the milk exosome then deesterify the peptide, which it reinitiates the charged component. It's now a negative charged peptide held within an exosome in which the membrane is negatively charged, and it cannot get out. And that's shown here in our second one, where the peptides will slide in through the membrane. They're now deesterified and they can't get out. Exosomes then are orally administered. They bypass through where they need to go. They get to the cells. They merge into the cell membrane, and they deliver their contents directly to the cells. So I was debating how to show the efficiency of this. We get about 30 micromolar on average. And I was gonna show some HPLCs, but that seems a little boring. It's just lines on a page. And I figured if we're going to show lines on a page, I'll just show something that has a little bit more weight to it. So what we've done, and this is about 9-10 months ago actually now, we worked with Dr. Stefano Toldo and Dr. Antonio Abate, who are experts in giving mice heart attacks. Indeed, they're the group that we worked with on our targeting studies. The difference here was that Dr. Toldo and Abate induced an LAD-induced ischemic reperfusion event, so the Widowmaker heart attack, and then so that they induced the ischemia, wait 20 minutes, and then reperfuse. After reperfusion, we administered 100 microliters of our peptide-loaded exosome, that's termed Exolacta, it's a placeholder name, and so 100 microliters, that's about 3 meg per keg at about 75 micrograms in there, with about 5E11 particles being administered to the mouse in this situation. At 24 hours after oral gavage of our treatment, the mice were then sacrificed and the hearts were measured for scar size and heart function. And what we found was a potent ability for milk exosomes loaded with therapeutic peptide to reduce the scar size in an ischemic reperfused mouse heart by over 60%. Now, this is a proof of concept for orally administered milk exosomes loaded with a pharmaceutical agent to deliver directly to the organ of interest and the organ that is currently undergoing, in this case, a dramatic injury. And we can protect those organs from massive organ failure. Now, this extends far beyond the cardiac research. Like I mentioned, this was 9 or 10 months ago. We're now moving this out, and we're expanding this as this is a drug delivery platform. You can isolate milk exosomes in the method that I instructed earlier. You can then load them with a drug as long as it can fit physically within the exosome by shielding those charges and using glass transition temperature incubations, and then orally administer this combination therapeutic and translate the ability of, in our case, peptides, which degrade in the body, the bioefficacy of this therapeutic within the body directly to the organs of interest. So basically what we've created to this point is a bioactive wound targeting drug delivery system that for cardiac therapies could be revolutionary. This can then be lyophilized, and it can sit at room temperature at this point, I've received 0 negative results, and we're out to 9 months. So we're going to keep taking it out to about 3-4 years. But to date, we've seen no degradation of bioactivity of our lyophilized samples. We are currently in preclinical validation. However, PureTech Health, who is doing something somewhat similar to us, is currently in clinical trials for their uses. Moving forward, Tiny Cargo is actually kind of switching gears. So the cardiotherapeutic is great, but we also have a peptide that is highly radiotherapeutic. And as of current, there are 0 FDA-approved medical countermeasures for the exposure to high-dose ionizing radiation. And as of current, the geopolitical situation necessitates that we have something that can treat against those effects. And so we are actually currently partnered with the Blue Knight Group, which is a BARDA JLabs co-venture. BARDA is the Biomedical Advanced Research and Development Authority in the United States, and they are behind our CBRN portfolio, or the chemical, biological, and radionuclear threats. And so we are currently meeting with them. I think actually in exactly 1 week, I'll be in DC meeting with Blue Knight and BARDA in order to implement the path forward to advance our medical countermeasure. And in industrial processing, obviously, we're working on it, but I also want to shout out Ken Marcus from Clemson, who's advancing a very innovative method of purifying exosomes from milk as well. So I believe that is it. Yeah, so thank you all very much. I appreciate your time, and I'm looking forward to the question and answer in a bit.
Thank you, Spencer, for this very interesting talk. And it's always interesting to be exposed to the industrial aspects of things. And your talk actually took us all the spectrum, from dry benchtop practices through scaling up all the way to biological efficacy of Milky Vis. So thank you so much for that. I would like again to take the opportunity to thank our audience and for the great and interesting questions that you were posting. And please continue to do so in our Q&A box. Of course, we will address these questions to the speakers during the Q&A session at the end of the workshop. And I'm very happy to introduce our next speaker, Steve Hicks, physician scientist at the Pennsylvania State College of Medicine. Steve, please, the floor is yours.
Thank you. Hi, everyone. Really excited to be here today and share with you all the work from my lab on looking at milk microRNAs and their potential impact on health in breastfeeding infants. I'm a general pediatrician at the Penn State College of Medicine. Some of my other work with microRNAs involves saliva microRNAs and their use as diagnostics. And in that role, I serve on an advisory board for Spectrum Solutions and Quadrant Biosciences. But today I'll be talking about my work in milk, and that is not industry affiliated at all. So as a pediatrician, I always share with families that breast is best and breast milk provides the optimal nutrition for their developing infants. I cite studies about how it impacts rates of otitis media, gastroenteritis, and upper respiratory infections, reduces risk for allergies and autoimmune diseases, and talk about how it might enhance immunologic function. But the truth of the matter is that I don't really understand, and I don't believe that any of us do, how this actually happens, at least on a long-term basis. So I think we understand how IgA and lactoferrin and lysozyme and other components of milk, as they're being consumed, can impact health positively. But it's hard to explain how breastfeeding for 3 to 6 months could have sustained positive benefits on infant health a year or 2 years out after breastfeeding. And so I began thinking about how microRNA might play a role in this, because that's my emphasis of study as a physician scientist. So we know that of all the human body fluids, that breast milk is one of the richest sources of microRNA, just behind saliva and seminal fluid. The microRNA in breast milk is packaged largely within exosomes, but you can also find lots of it protected just within lipid bilayers in the lipid fraction of milk. And we know from lots of really interesting research in animal models that it's capable of surviving the enzymatic milieu of the gut. We also know that there's a lot of targets of microRNAs involved in immune function. So it makes sense that this is a potential marker for, or molecule for, transmitting sustained health benefits, especially when considering that there are no human microRNAs in formula. So the current state of knowledge regarding human milk microRNAs was really nicely summarized about 1.5 years ago by Tingo and colleagues in a Frontiers in Immunology paper. And basically, just to kind of put that entire paper into just a few bullet points, we know that milk microRNA originates primarily from epithelial cells. We know that microRNA profiles are fairly consistent across different cell fractions, but that sample collection, storage, and other processing variables can have a big impact on the microRNA that you measure in a sample. By and large, however, the majority of literature out there is descriptive in nature when it comes to human milk microRNAs. And a lot of it is pilot data. So I'm talking studies of 20 women or less. And that really is a major barrier to us understanding how this helps infant health, right? We need our large-scale studies that connect the milk microRNA content with infant health profiles over time. So the challenges that I see as a pediatric physician scientist are that we don't have these kind of large studies which look at the content of microRNA over the course of lactation and how that content could be impacted by maternal variables such as smoking status or age or parity. We also don't know for sure that milk microRNA has any impacts on infant health. There's good data in animal models, again, to show that it does, but to my knowledge, there's very little out there tying human milk with infant health outcomes. And if so, which microRNAs are responsible, right? Because there's hundreds in milk. And then finally, we don't have a great understanding again, in humans, how those microRNAs might modulate infant biology, especially when you start to consider all the different environmental variables that can contribute to health outcomes, like allergies and infections. So to try and address some of these deficiencies, we designed this study called Bioactive RNA in Maternal Breast Milk. It was funded by the Gerber Foundation, and it ran over the course of 3 years from 2018 to 2021. And our goal was to investigate associations between immune-related microRNAs in breast milk and infant health. So we actually had pretty specific hypotheses that we came up with based on reviewing the literature in animal models. We pinned down 3 immune-related microRNAs that we knew to be present at pretty high levels in breast milk that had been shown to modulate either T cells in cell culture models or even health outcomes in animal models like rats. And then we hypothesized that these immune-related microRNAs would actually modulate some cytokines or inflammatory markers in the infant oropharynx. So this study was a longitudinal cohort study of 200 mother-infant dyads. We actually enrolled 221 to account for dropouts. The inclusion criteria were a maternal age between 19 and 42 years, term delivered infants, and at least an intention to breastfeed beyond 4 months. We excluded maternal infant dyads that could not breastfeed either because of a maternal or neonatal condition, or a plan for infant adoption or to move outside of our medical center. And that was mostly just so we could be sure that we were able to track these infants out to at least a year to determine their health outcomes. So we collected milk from the mothers at 3 time points, at 0 months, 1 month, and 4 months. And then we collected infant saliva at 6 months. And we layered over top of that a whole bunch of standardized surveys that allowed us to collect not just basic medical and demographic information, but also the IFP for infant feeding practices, the SCORAD, which is a standard tool for looking at atopic dermatitis, the DSQ, which is a dietary screening questionnaire to look at maternal diet and how that might impact milk levels. At 1 month, we collected the NSLAH, which is a national survey of lead and allergens in housing to look at environmental impacts. And then zooming out to the 12-month time point, the EISLWQ, which is looking at wheezing-related outcomes, upper respiratory, and other infection-related outcomes. So we screened almost 2,500 dyads for inclusion in this study. We approached 359 who met criteria, consented 221, and then got at least 1 milk sample from 192 of those dyads. That included 176 samples right at the time of delivery or within the first week of delivery, 178 samples 4 weeks out or 1 month out from the delivery, and then 146 samples at 4 months. And that drop at 4 months is due to mothers who gave up on breastfeeding earlier than planned. So this is a very busy table, but what I want to point out is that essentially there's no change in age, race, or ethnicity across those samples at 0, 1, and 4 months. Our cohort ended up being slightly overweight based on BMI. They had rates of gestational diabetes right around 11%. The majority had breastfed before and breastfed more than 4 months, but almost 40% had never breastfed before. We also looked at factors like milk maturity, collection time of the sample, and how that might impact milk microRNA content. And you can see that there's minimal change in that over the 3 time points. And then we used the DSQ, as I mentioned, to look at maternal diet. And that includes basically dairy, calcium, fruit, sugar, and vegetable intake for mothers. Okay, so first challenge, again, just to reiterate, is that we have no large studies prior to this that I'm aware of human milk microRNA patterns and what factors really affect them. And so to address this, we did longitudinal RNA sequencing on over 500 milk samples from this study. That allows us to look at every known human microRNA in the sample and then compare how medical and demographic factors might impact it. So if I had known that I'd be invited to speak at this consortium, I would have focused explicitly on exosomes, but hindsight is 2020. So we did not. We actually just measured microRNA straight out of the lipid fraction. I do have a little bit of data here to show that at least for 2 of the really most common microRNAs in milk, the levels within subjects are fairly comparable across lipid and exosome fractions for us. So we used a Norgine exosomal RNA purification kit and a NextSeq 500 for RNA sequencing. And what we found is that milk microRNA is surprisingly homogeneous in human milk samples. So of the over 2,600 known human microRNAs, we can detect almost 1,800 of them in at least one sample. There's 238 microRNAs that we classify as being consistently present, meaning they've got a raw read count of at least 10 and at least 10% of the samples. But really, the content of human milk microRNA is made largely of about 11 microRNAs. So these 11 microRNAs in this bar graph here all the sample. Different factors might affect these 11 microRNAs. So first, looking at milk maturity, meaning across the 0 month, 1 month, and 4 month time points. It turns out that maturity has a pretty significant impact on all 11 of these microRNAs. And I'll show some spaghetti plots to kind of visually demonstrate that in a second. But really, not a lot of other factors demonstrating a huge impact on these 11 microRNAs over time. So maternal dairy intake affects about half of them. And then miR-146b5p, which is the most abundant human milk microRNA, at least in our hands, is impacted by calcium intake and also maternal gestational diabetes. So some microRNAs go up over time, some go down when it comes to milk maturity. Here's 4 examples. So you see at the top, miR-181c, from 0 to 1 month to 4 month makes up less and less of the milk microRNA content over time, as opposed to miR-30d5p, which actually proportionally goes up over time in maternal breast milk samples. So the longer a mom feeds, the larger proportion that microRNA is going to make up. Although I kind of just showed you for those 11 really common microRNAs, not a lot of factors that seem to affect them. That's not the case for all microRNAs, particularly some of the less abundant microRNAs, like miR-196a5p, shows an effect of maternal parity, maternal smoking, and maternal age. So you can see this microRNA, as a mom has had more and more children and breastfed presumably more and more times in her life, it makes up a greater and greater proportion of the milk microRNA content. It also is higher in the milk of mothers who are not smokers and higher in older women. Okay, so the next step for me is that as a pediatrician is to figure out how each of these individual microRNAs might be impacting infant health. And the way that we did this was to kind of devise this, as far as I know, fairly novel approach to try to quantify how much milk microRNA the infants in our study were actually swallowing or consuming in the first 6 months after delivery. So the way that we did this is for each infant, we had a milk sample at 0, 4, and 16 months, as you recall. And we came up with an equation which pairs the milk microRNA concentration with other factors that we'd collected about how much milk each child was consuming day over day and how many days they continued to breastfeed for. And so what we can do is multiply the milk microRNA concentration at time point 0 with the volume of milk consumed in that period from 0 to 4 weeks, and then multiply that by the duration of days in that period that they were actually feeding for. And so for each individual microRNA, the 238 or so really kind of consistent microRNAs, we went and calculated for all the infants in our study, how much did each infant actually consume. So now we're looking at microRNA consumption in terms of milligrams over the first 6 months. And this is very different from most microRNA studies you're going to look at, where microRNA tends to be reported or visualized as a ratio. So a microRNA of interest divided by some sort of housekeeping gene, or reads per million on a sequencer. where here we're actually looking at the milligrams of that particular microRNA consumed over time. And so what you can see is that for really common microRNAs like 146B, 5P, they're in the middle, an infant might be consuming around 40 to 50 milligrams, in the first 6 months, whereas less common microRNAs like miR21, which was part of our initial hypothesis, most infants are consuming less than a milligram. And so then what we did actually was divide up our cohort into milk from mothers whose kids went on to develop atopy, which is a medical word for 3 different allergic conditions in the first year, and then milk from mothers whose babies did not develop atopy. And you'll see here that one of these 3 that was part of our initial hypothesis, miR375 3P, is actually higher in the milk of mothers whose babies did not develop atopy, so it's protective against atopy. We built that into a linear regression model, which controls for sex and race, delivery method, daycare attendance, environmental allergens, and passive smoke exposure, all factors known to impact rates of atopy in babies. And that odds ratio remains significant. So basically, having miR375 in your breast milk cuts risk of atopy in half for babies. Interestingly, it's also inversely related to maternal BMI. So meaning if you can actually encourage mothers to lose weight, you can have a positive impact on their miR375 3P levels. So that could also positively impact infant atopy outcomes. In terms of upper respiratory infections, we can perform a similar linear regression control for factors like daycare attendance, the number of children in the household, and other things that can really have a big effect on whether a baby has a lot of upper respiratory infections in the first year or not. And even after controlling for all those environmental exposures, we still see some pretty large effects of particular immune-related microRNAs. So miR21, for example, is protective against the number or frequency of upper respiratory infections that a baby has in the first year of life. The last challenge for us is to sort of delve into how these different factors might impact the kind of biology or physiology going on within the infant. And I mentioned earlier that we collected infant saliva. So we actually ran multi-omic analyses on all these infant saliva at 6 months. We measured cytokines, the saliva microbiome, and saliva transcripts that were related to immune function. And then we can kind of look at how the consumption of those 2 microRNAs I just showed you, miR375 and miR21, might actually be impacting the physiology going on in the infant's oropharynx at 6 months. So it turns out that miR375 reduces the activity of proteobacteria in the infant oropharynx at 6 months. And it does that through reducing transcription of the TAB2 messenger RNA. And TAB2 actually boosts proteobacteria. So proteobacteria, for those of you who aren't in microbiology, is a group of bacteria that includes negative anaerobes that cause E. coli or salmonella. And TAB2 transcript regulates TGF-beta and IL-1. miR21, in comparison, does not have a direct impact on CXCL10 cytokine protein levels in saliva, but it does indirectly affect them. And it does that by modulating fusobacterium activity. It blocks fusobacterium activity, which has a blocking effect on CXCL10 cytokine production. Fusobacterium are gram-negative anaerobes that are implicated in peritonsillar abscesses and gingivitis. So now we have a good harness on sort of what the actual milligrams of consumption of milk microRNAs are in the average infant. We've pinned down particular ones that seem to be related to health outcomes attributed to breastfeeding. And we have some potential mechanisms for how those particular microRNAs might be positively impacting infant health in the oropharynx. Next, we plan to try to set up some collaborations to use animal models to look at efficacy and dosing of these particular microRNAs. and then determine if we can actually modulate them within human breast milk by maternal diet or exercise interventions. So just to conclude, pretty few microRNAs, just 11 that predominate within milk. That composition changes over time. Maternal factors, particularly diet, can impact that composition. We've identified miR-375 as being associated with likelihood of infant atopy. It's protective. And miR-21 as being protective against frequency of upper respiratory infections. This is my lab who all participated in the generation of this data, and I'll wrap it up there. Thank you very much.
Thank you so much, Steve, for this fascinating talk. And again, reminding all of us how important maternal factors like age, metabolism, etc., in the milk that is being produced to the baby, and including how mighty exosomes are in this fluid. So for our audience, please do not forget to submit your questions in the Q&A box. We're still harvesting these questions, and we will address them to our speakers in the panel discussion that will come up. We will have a 10-minute break before our Q&A panel starts. So we will meet here in 10 minutes exactly. So since we're talking about multiple time zones around the globe, I will not tell you what's the hour here now, but we will meet in 10 minutes right here, and we will get back to you. Thank you so much. So welcome back, everyone, to our final part of today's workshop. And I think it will be safe to say that this is the most exciting part, where we have the privilege to engage with our distinguished panelists and address the questions that you have posted and discuss it with them. As a reminder, the main objective of today's workshop is to share and discuss the challenges of understanding exosomes, their composition, analytics, physical and chemical properties, and of course, their efficacy. We aim to explore methods for isolating and scaling up their production, as well as the regulatory challenges that we may all face. Our hope that by open discussion about these topics, we can collectively advance our understanding in the field of exosomes. So we have many questions to all of our speakers, and I would like to start, I think, with one of the interesting ones that were posted by Paris and Glid, asking about the bioactivity of milk from other mammals in terms of their effect on coagulation. So Renk, you actually showed that cow's milk is not as active as breast milk, but Per continues to ask if EV can go through the digestive system and still stay bioactive.
It's a very interesting question. So what I did not show in my presentation is that if you collect normal human blood, so for normal healthy humans, you will never find any tissue factor activity there. So you only do see tissue factor sometimes when you have patients with cancer, and sepsis, and COVID patients, etc. And then it's either derived from, for example, a tumor or from inflammatory cells. In principle, if tissue factor enters your blood, you have a really increased risk of venous thrombosis. For sure, if tissue factor from, let's say, bovine milk, well, it's not there, but if it would cross-react with human factor 7, you would be at risk for thrombosis. Maybe it's good that we mostly drink bovine milk and there's no tissue factor there, but yeah, it would be very dangerous, for sure.
Okay, so I will continue talking a little bit to all of you about the safety and the aspect of using different mammals as milk from different mammals as a source for exosomes. So a question from Hank Bovenhaus, asking Spencer, but I think that all you can answer. Spencer first. It was indicated that EVs are evolutionary conserved. Consequently, one might argue that milk from one species consumed by another species will result in a mismatch, wrong singles transferred to the offspring. Isn't it better to treat milk and in this way destroy EVs signals?
Yeah, it's an interesting question. I think from our view, the exosomes from cow's milk that we've used in our mouse experiments have shown extremely high levels of bioactivity, and we haven't seen any issues with toxicity. We performed some baseline safety and toxicity tests in mice where we use a scaling dosage, and we found no effects of very large doses of exosomes in mice. My only experience is with utilizing cow's milk exosomes in a mouse model. So we are limited by that. And I think some of the other speakers probably have a bit of experience that they can talk on here. But from my perspective, the evolutionary conservation of EVs and the production of milk is pretty far back in our evolutionary chain. And from what we can see, I mean, I didn't actually highlight this and I should have, but the addition of 100 microliters of concentrated exosomes to a mouse that's getting a heart attack, and this is just the exosomes, not the therapeutic cargo that we included, reduced the scar size by about 30%. So even in this mouse model, in a bizarre situation of ischemia reperfusion, the cow milk microRNAs do appear to be both highly bioactive and helpful. And in our safety toxicity tests, we've seen that they have little, if no, toxicity, even at 10, 20 times dosings.
Maybe I can add something to that. Yeah, the point is, if you look to the most conserved microRNAs in these species in milk vesicles, you can see that a lot of these microRNAs are identical, not look alike. They are really identical. So meaning that there is not a difference between, if you look to a microRNA activity, whether you have the bovine EVs or the human EVs. The question is of course, which cargo is doing the job? You cannot say that there's only one microRNA doing the job if you have all these other cargoes around there. So it is a very good question. But from microRNA point of view, you can easily explain things like if you see this this cross-species activity being so efficient that is 100% conservation between the most abundant microRNAs. Not saying that that are the most important microRNAs. That's my other point, but yeah.
Okay, thank you. Maybe following up with a question of our own IMGC Sac membrane, Dr. Lilia Strickland, who's asking, Steve, have you looked for or found any association between specific milk microRNAs and a negative health outcome?
Yeah, so that's not something we've gone after just because there's not really funding to go out and show that milk microRNAs hurt children. But I will say that, right, when we're looking, we take an agnostic view when it comes to looking at associations between microRNA consumption and a positive health outcome, like having less upper respiratory infections. And although I only highlighted the microRNAs that had protective effects today, we do see others that show possibly like non-protective effects or negative effects. So drinking more of a particular microRNA is associated with having more upper respiratory infections. That's not something that we're particularly like searching for, but we do uncover it when we're doing these kind of studies. So,
okay.
Yeah, and I think that maybe also to come to that point back, like you can search specifically for things, but that is where I brought in that you can also let the machine learning and artificial intelligence search for you. And for example, like where we do look to the differential expression of microRNAs between the milk vesicles from allergic and non-allergic mothers, I'm not saying that they have positive or negative effects yet on the children, on the offspring, but you do see differences. There are differentially expressed microRNAs there.
That is interesting. Again, a follow-up question on exactly these your answers. Maybe going back also to Steve. Can the findings of atopy affect or mothers' microRNA affecting atopy at the children? Can it be confounded by genetics of the mother or predisposition of the mother influencing her infant? How can you be sure that it's the microRNA or the EVs from milk?
That was a really excellent question. I saw it in the chat. So the answer is yes, it absolutely can confound it. And so that's why just showing the box plots that I showed initially are insufficient to draw any conclusions, right? So just saying like this microRNA is higher in the milk of mothers whose babies didn't have atopy, and I did a t-test to show it, like that's not helpful. So that's why we do the more like in-depth linearized regression models where we're controlling for all these covariates. And one of the covariates we're controlling for is maternal history of allergy, right? And so by doing that, we're essentially controlling for maternal genetic background, which is passed on to the baby.
Okay. And another question concerning how the mother transfer her inputs, so to speak, not directly through EVs, is for you, Rank. How can you distinguish the effect of suckling, and maybe the tissue factor can come from, you know, the nipple being hurt through suckling, or some kind of a pressure or vacuum that is implied during the suckling or the eating session, and how can you be sure that this is not also affecting the efficacy of milk or milk EVs on coagulation or other factors that are being affected?
Yeah, it's a good question. I think that the question in general is that why is tissue factor so abundantly present in normal human body fluids, including milk?
Right.
And to be honest, I think we only have an hypothesis, and that hypothesis is that that body fluids help to protect against microorganisms entering the body. So they provide a sort of hemostatic envelope, meaning that around your blood vessels you have tissue factor, that these body fluids provide more tissue factor. And just to give you an example, if you take the fastest working snake venom, that's an enzyme that you can add to human blood, normal human saliva has exactly the same biological activity. So the ability of your saliva to trigger the clotting of your blood is just as potent as the fastest working snake venom. And milk is even more active. Milk is for us, the sky is the limit. I said we can dilute it 4,000-fold, and we can still easily measure that human milk has the ability to induce clotting. Why? Well, I think it is double. I think it is protection. So also protection of the mother when you have nipple bleeding. I think there it will also for sure be helpful. But of course, these vesicles with tissue factor and factor 7 also come into the gut of the baby, and I wouldn't be surprised that they also have that they regulate, or they help to regulate the tight junctions of the epithelium. I think that's what we're trying to prove now. So yeah, I think it's a double. It's all about protection in the end. So you can say that tissue factor on the one hand helps it helps to tighten holes. So when you have a wound, it will close the wound, but it may also be helpful to regulate tight junctions.
Okay.
And in the end, both are aimed to keep bacteria out and microorganisms.
Thank you. Okay, so next question is for Marka. And I think I saw that you answered it on the chat, but I think this is a very important question that I will be happy to have everybody listen to your answer. This is from Janet Williams about the use of milk EVs from frozen milk. So I know that you mentioned the effect of pasteurization and homogenization on the efficacy of milk EVs, but she asked specifically about freezing. And I think it's very important when women actually freeze access milk in their freezers and then thaw them and give them to the baby. Do you have any input on that?
Yeah, I think that was exactly an important point. When we started our study on milk EVs, we started with all the very nice biobanks that are available for milk, and it turned very rapidly out that they were not very useful for our study in doing really trying to find out about vesicles naturally present in milk, what they contribute to the immune modulation and to the crosstalk with the epithelial barrier. And that was because when you collect your milk, also as a mother, you just freeze directly your milk at -20 at home. And in the lab, maybe at -80, and that was also how it was biobanked. But all the cells were still in there. And that gives an enormous background in it. Because these cells, they fall apart very rapidly, and they fall apart in all kinds of vesicles. So you do not know what is derived from cells that is an artifact of the freezing and what are the natural vesicles. So when we started our study, we started a new biobank, and then we first started, for example, to store the milk at 4 degrees, set the mother to collect the milk, stored at 4 degrees, so that we could do the first step to defat the milk. And then we already saw that directly, also at 4 degrees, a lot of these cells were dying rapidly. And it took a while, but we found that bile salt-dependent lipases that are present in milk, that they lose their bile salt dependency when they are at 4 degrees. Oh. So actually the cells started to die very rapidly, also at 4 degrees. So we made a horrible protocol that the milk after collection should be kept at body temperature and should be processed within 1.5 hours max with the first steps. And that was the 2 times 3000 G spin to remove the fat. And at that point, after that, we freeze the milk, the milk supernatant at -80. And there, so that is our biobank. So directly spin out the cells. Of course, this is not a guideline for mothers. And I was struggling with that because it is also dangerous to say like, it is not good the way how you do it, because I am convinced that giving your own breast milk and if you use a breast pump and have it at -20 is still much better than formula milk, but it looks different, looks completely different. Yeah.
It's an amazing, like new, not concept, but definitely one of the things that we wanted to see is the challenges that all of us are facing during very initial steps of science, of understanding the factors that affecting EVs, and collection method. It's a key in this process and in these studies. So again, a question for you, Marka, asking about the different methods for casein micelle deaggregation that you show that affect EV's properties, and the question is, have you examined those effects on proteome and microRNAs? So
yeah, it's a very good point. And I think it's always, if you compare these different methods like the EDTA we did, the sodium citrate and the acidification, you always have such readout systems that you that you use the NTA, so the nanoparticle tracking analysis, protein concentration, phospholipid concentration, etc. Sometimes, so the more things you test, the more differential effects you find. And that is something which is really important to keep in mind. If you, for example, are only interested in tissue factor, then you can say, I look to tissue factor. Is it affected? Is the blood clotting affected? Yes or no? You can tell something on tissue factor, but not on the vesicle integrity or on other things. So that is where you always need to tailor exactly the question, like what do you want to address? The point that is asked, like, did we do the full proteomics analysis and the RNA analysis after these different reasons? We didn't do it, but it is a very important point. We look to the big parameters, but it is definitely, if you want to tell something on it, you should do it. You do, and then not just 10 abundant microRNAs, you should do the full sequencing.
Definitely. I want to ask, following your input on the protocol, the collection protocol and separation protocol, and how differences they might be when you're freezing or just chilling the milk prior to processing. I would like to ask Spencer, because you mentioned cholesterol, and cholesterol has it's a very unique molecule, and the phase separation it's a very tricky molecule. Can you comment on maybe on the separation protocols regarding the cholesterol content or the special membrane? Because I'm figuring it's not really similar to the regular epithelial cell membrane composition.
Yeah, so the processing that we are currently using, we put it at 37 when we do our chelation, and that was found to be extremely effective at dissociating that casein from the extracellular membrane. But these change throughout temperature. So at 4 degrees C, it's much less effective, much less, because at 4 degrees C, that association is going to be much higher, and that's driven by these cholesterol rafts, which get extremely rigid at lower temperatures. And we haven't done studies on the source of this, but we have found that freezing at very specific steps is okay, which I believe Marcus said, but at other points it's not. And we do see that it's very interesting, and it does come back to the biphasic membrane and how these are going to be interacting with each other and with the other components at that period of time. And so I think it's been a while since I've looked at this, but I believe cholesterol makes up to about 40% of a traditional exosome or extracellular vesicle membrane, which means that you have to take this into consideration when you're doing your storage. And that really, we believe to be the driving force behind the requirement to have a cryoprotectant in place when we perform our lyophilization. I briefly mentioned this in there, but without that cryoprotectant, the lyophilization, you result in about 10% yield. And with that, you get about 9,500. And that's going to be driven by that kind of biphasic membrane, as you mentioned.
Will you have problem to isolate, not problem, but will you have challenges of isolating EVs from milk of different cows from different countries? So for example, in Israel, there might be higher cholesterol content in milk compared to the US or other European countries where cows are pasture. Usually Israel, they're fed concentrate diet. There might be differences there.
I would love for everyone else to comment on this, because up to this point, unfortunately, we've been getting our milk from a producer here in Virginia on the East Coast. We've worked with a small raw farm a little bit further south. But in 2 weeks, we're going to New Zealand to work with their cows. And I have no idea I've done different species. So we've done goats and sheeps and cows. I did work with a group doing human milk here as well. So across species in this area, it seems to all work fairly well, but it's a consideration that I've been kind of pushing off because I really hope there isn't some massive difference between the products across the globe, but I think there will be. And I think I'd just like to hear what you all have to say, because I only have a very brief experience with East Coast cows. So
anyone?
Yeah, so we mainly concentrated, of course, on the Holstein Friesians, and but it is a very good. It is really a very good point to look into the composition of these vesicles, and especially also the membrane. And so we did not do the lipidomics on these vesicles, but we do quite a lot of lipidomics on other extracellular vesicles now currently in my lab. And there you can also see that, there are also big differences between subsets of vesicles also in the protein composition. And that, for example, if you analyze the lipidome of the vesicles isolated from synovial fluid of a healthy joint or of an inflamed joint, we can make an inflammatory lipidome profile that is different from the healthy lipidome profile. And I think there is really a way to go in investigating the membranes, because they now, if you look to the vesicle papers, they are always drawn like a very homogeneous envelope. So I like also Spencer's talk because that is not at all the case. And if you now do lipidomics, you said these are such a stupid schematic that you have these proteins there and the metabolites, and that there's a very homogeneous lipid bilayer, which is not the case. I think this question and also the approach you took, Spencer, to maybe use it for loading the vesicles in another way. I think there we should take much more advantage of all the knowledge that is present in the milk area, because people did study a lot of membrane work in milk. And I think there is the advantage in bringing the milk field together with the extracellular vesicle field, in that we can, from the milk, people learn a lot about the lipid membranes.
Yeah, and also about the composition and the effect of diets, cows, mothers. I'm sure there are a lot to learn there. We have several questions about the source of milk EVs. So this one is for you, Steve, but we can continue with and address these questions to other speakers as well. Martin van Herenwijnen is asking, how much are you sure at the source of milk EVs that are actually coming from the epithelial cells? Because there are a lot of immune cells as well. And can you distinguish between the ones that are coming from the epithelial cells, from the ones that are coming from elsewhere, maybe also from the stroma of the mammary gland itself, so fibroblast, for example? Please.
So yeah, I can, and I can't. I'm not probably in the best position to answer that question or comment on it. We took in our own work an agnostic approach to this, which was just, these are all the microRNAs in a human milk sample. They're hitting the infant oropharynx. We want to know which ones are impacting health, regardless of what cell they originated from or what they're encased in or protected in. But I think that there's truth to that. When I said that they came largely from mammary epithelium, that's a citation of someone else's work, not my own. So I'm sure there's probably, I'm not a milk biologist, so there's probably conflicting data out there or data that they come from other places just as much. So, but again, that citation in my own slide deck was in relation to microRNAs, not extracellular vesicles. So just to clarify.
And also the question, is it important? Maybe Marka or Rank can answer that. I mean, milk is a fluid that comes from true from the mammary epithelial cells, but also from the blood. For antigens, for example, coming in colostrum, nobody will argue that they are not appropriate there, right? They are part of the bioactivity of milk. So can we say something? Maybe.
Well, I think in a few studies, probably Mark has done it as well. In principle, if you have cell-derived vesicles and they expose antigen of the parental cell, you can identify these quite often by using flow cytometry. So then you're able, from each single particle that you detect or single vesicle that you detect, you can tell whether for example, whether the vesicle is from epithelial origin or not. The problem is that many vesicles have only a few epitopes. And I think that, let's say, all vesicles under 150 nanometer are still really tough to tell precisely what their cellular origin is. So for the larger vesicles, for sure, it's possible. For the smaller ones, it's difficult still. But yeah,
Oh, but I think
it's developing fast.
Yeah, but I think this is so we have a paper which is still under review now, but it is for that we use the bead-based assay. And so you capture on a certain antigen, for example, CD3, which is really a T cell derived antigen, which is not on epithelial cells. You just capture vesicles with CD3. So you know they are not from an epithelial cell. And also, if you then do the detection not with what the manufacturer is saying with a PAN tetraspanin detection. So normally you use the mixture of CD81, CD63, and CD9 to detect all vesicles, but we did also the individual tetraspanins. So you can see fingerprints back from certain cellular markers with certain tetraspanins, because not every tetraspanin is on every vesicle, but so also if you do not use the single EV analysis with flow cytometry, but do the bead-based analysis, you can already get a lot of information on it and you can trace it back to the cells of origin. So that it is for sure that it is a very heterogeneous source.
Okay. We have, again, questions in the same area, more or less. For example, Sally Ibrahim asked, could cows with subclinical disease like mastitis or endometritis affect EV's efficacy? And I will add composition for using in the industry.
I'll just briefly say any situation going on in the cow is going to impact its EVs. If it's healthy, if it's injured, if it's diseased, it all has an impact.
Okay, but maybe still no toxicity effect is currently.
I think the question said that about 5% of all cows are right. So in that case, just from my experience, the cow milk that we get is pooled among a number of cows. They're all A2 jerseys, but it's pooled. So let's just say 5% about have this. Overall, then we've seen no issues with toxicity or safety. But that's making a few assumptions, and I'm not sure about the rates of this at Homestead. But I would imagine that especially if you're pooling milk, if you're taking it from one cow that is diseased, I think you'd see problems. But once you start pooling this and the rates go down, I would imagine that would be reduced.
Yeah, I fully agree with you, Spencer, that if the farms are monitored for the cell numbers, etc. So now it's really very good monitored, and you can have some subclinical disease cows indeed there. But if it is in the tank milk, and you have quite a big group of cows there collecting to the tank milk from which you take your sample, it will be diluted out, and of course you should check it. But I think overall it is not such a problem if you are not using individual cows.
Okay. So the dilution effect is important here.
Yeah.
The greater the farm is, the larger the farm is, maybe. Another question from Janet Williams. Maybe Steve and Marka, maybe you can answer. Have you investigated longitudinal changes in the content of EVs across lactation and or between colostrum and mature milk?
So Steve.
Yeah, so unfortunately, we have not looked yet at EV composition, only at microRNA composition. So I can say that changes dramatically from early milk to 4 months out. But as it relates to just EVs themselves, I can't say from our own work at this point.
Yeah, and I had the problem that from ethical points of view, we could not get, of course, the very early milk in such quantities to do a very good comparison. But now we first made a whole biobank with goat milk in which we made this whole studies. So, but we need to analyze it. But that is a lot of work to isolate with this analytical protocol all the vesicle samples, and then afterwards do all the microRNA, so the whole sequencing and the whole proteomics. But it is very important to do it.
I just can maybe mention how important it is, especially relating to your studies, Mark, on the reepithelialization and the gut barrier. So all mammals, if I'm not mistaken, maybe I will not generalize, but surely ruminants and humans are born with the leaky gut, so to speak. So maybe EVs in this early on has a large impact on maybe expediting this or making sure this process happens in the right rates.
Sorry.
Yeah.
Oh, no,
So
you're
we
fine.
started to work with the pigs because the composition of the milk of the pigs is more similar to human. And but we had some problems with milking the pigs daily because they are very smart and they became quite aggressive. So there were some logistical problems with students.
Yeah.
And so I will say on that note on the barrier function in the gut, we in one of the more recent experiments that we've done, we're looking to advance a radiotherapeutic medical countermeasure. And one of the organs in your body that is heavily impacted by radiation is the gut. It's highly regenerative, and you get large breakdown of the crypts where the intestinal stem cells are, as well as the barrier there. And when we gave a lethal dose of radiation to test the effects of our therapeutic, in that you have a vehicle control, which is milk exosomes in this case. When we gave these mice milk exosomes, we then took the GI tracts out and had those graded by verified histopathologists, and they graded the milk exosome group as equivalent with the non-irradiated as far as the gastrointestinal tract was concerned, which to me, I still can't quite wrap my head around that. That the ability there, and it was primarily in crypt regeneration, which means that even in a situation of a lethal input to the gut, just the addition of milk exosomes was able to recapitulate that healing to where it appeared as if it was non-radiated. So there's some really, really powerful mechanisms there at play.
Excellent. Okay, let's continue from a question from Jennifer Smilowitz. What is the most important target of EVs to bring them to the clinical settings? Maybe Steve Rank can start answering.
Steve.
Yeah, so I think that first we need to know, from my standpoint, when I just compare this to drug development, right, we need to know what the range of doses is that we have to be exploring. So essentially, like, what is the least amount that infants see, which is 0 for me, and just in my area, which is like infant exposure to exosomes, and what is the most amount? And so just defining that with descriptive studies, so you have a range, and then you can say, okay, this is the physiologic range that they see, And we know we have like improved health outcomes within that range. So then finding the optimal dose in there, I think like that's the first step in any drug development, is defining the safe range, or and then you can move on to see what's the most effective range. So that's essentially, you know, in our own work, that's why one of the first things we try to do is just a large scale descriptive study to say, this is the in milligrams, like a very quantifiable range of what a child would see in its first 6 months drinking breast milk.
But I think that the area, if that is what would be the most important area, for which you would like to use them, I think the point Spencer just made, the very strong regenerative effect and the anti-inflammatory effect, which is really unprecedented. Also, if you compare them with stem cells, the anti-inflammatory effect is so strong, and it fits with the proposed physiological role to control inflammation in the newborn in this new environment. So I think in these areas where you're looking to regeneration and controlling of inflammation, and then using the oral or nasal route, because also when children drink milk, there is also the nasal route is also a route via which milk vesicles can reach maybe the brain. That is also a very good point. And that is a new study we are doing now as well. And I agree that the dosing, and if you want to work with isolated EVs, that is very important. But if you just look back to the biology and stay close to the own mother's milk, then there are, I think, on much shorter range already things where we can apply minimally processed milk in the clinic, in the own mother-child diet.
So if I could probably give a bit of a different view of this, because we're not looking at this for infants and whatnot. From a pharmaceutical standpoint, the regulatory that is required for this is that we have to define ranges. So the size must be within a range, the lipid concentration must be within a range, the cholesterol, blah, 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 blah. All of these variables that define the vesicle, we have to standardize a range that these fall in, and it has to fall in that range 95% of the time. So the field as a whole, everyone working on milk exosomes, milk EVs, whatever you want to call it, the range of these needs to be standardized and characterized for these to be used in the instance that I'm talking about, in a pharmaceutical drug delivery system. So our focus when we do our lab scale isolation now, as we work to scale, is identifying those parameters, and continuously, every single time we run an isolation, we run a characterization of each of these components. And once the field is able to consistently generate within that range, that predetermined range, and it can be as wide or small as you want, but it needs to be consistent within a range, because these are heterogeneous. So yeah, that's my 2 cents.
But it really depends on the application. And
Yes.
So indeed, if you're saying I define the EVs as a drug carrier, that for that you need complete other regulatory things than if you have a complete other description of your EV containing therapeutic preparation.
Yep.
And I think that is important that we are discussing that within ISEF in the regulatory task force as well, that there are different routes that you can follow for making a carrier for drugs. You're right, there is this full description needed, and you can come up with the bandwidth, and it should be in there. But if you define the therapeutic entity in another way, there you can discuss about the description of your preparation, and you need to have the potency assays in place and the safety assays. The safety assays should always be in place, and you should have your potency assay. But then the other things, like whether there are other colloidal structures in there or not, so things like purity, play not such a big role, but it all falls in the design of your therapeutic preparation, what you are aiming at.
Yeah. So regulation is complex. No question asked. Again, Jennifer is keeping asking about the regulation, very smart questions. And she asked if you can describe barriers among the different regulatory agencies, such as FDA, EFSA, and maybe even USDA.
From my experience, the FDA currently classifies milk exosomes as generally recognized as safe, as they're a food product. But I have been indicated by a handful of regulatory experts that as soon as we make that case, the rug is going to get pulled out. So that's a work in progress for us at the moment. It is a food product, and that does classify it in a different manner. As long as it is appropriately sterilized, we see that the regulatory path as being fairly light as far as a pharmaceutical would go.
Does it imply also if you're putting different cargo into these vesicles? Yeah, still okay.
So so we're in the middle of these conversations, and it's tough. And I'll let Marka. She clearly, I think, has a bit more to add here than I do. When you don't modify the outside, they still classify it as being the same kind of interactions, and we're putting this inside. But that's an argument that we're making, and whether we're successful in that argument will be interesting.
Yeah, I think, Spencer, this is a good point, and the most important point is that so from the European part, I can only tell is that it is not so much that they make barriers or hurdles, but they don't know how to handle it. So we should be in a dialogue with them to really come up with proper definitions, and the two roadmaps I was just talking about is part of such a discussion. So it is also something we as scientists, also as very academic scientists, can contribute a lot, like what would be the proper description or not, and where are we going? And because if you go for these very pure vesicles and you lose function, which is often the case, that people say like, yeah, but we have a preparation that has a certain effect, but if I purify the vesicles from that preparation, not damaging them, but purifying, I see a much less effect. And that is because a lot of colloidal structures work together. Like Spencer said, K-signs are together with EVs at certain things. LDL particles bind also to vesicles. It's not just a contamination, it is also a physiological interaction. So for some of the therapeutic preparations, you need to have these colloidal interactions in there. So then it is not a pure EV preparation. And these things we need to pay much more attention already in the first design of the experiments, also in the academic experiments we are doing, and try to discuss together with regulatory agencies how we approach it to see, to get these roadmaps clear. But it is not that, I do not have the impression that they are against it, but often do not know how to handle it or classify it. But with the milk EVs, we have probably, as long if we are not going to administer them intravenously, but if we do that orally and milk EVs, I think we have the lightest path to get things done, but there is work to be done.
Okay, so you talked about the matrix when you were orally administrating EVs or milk EVs, and I have a question from Janice Zampleni. that you mentioned earlier, this group, right, Spencer, in your talk. How concerned are you about the degradation of milk EVs in lysosome upon internalization by recipient cells?
Yeah, it's a fantastic question. And the biogenesis of EVs and exosomes has been researched to the end of the world. But the uptake, from what I've found, is just very confusing. And there are 100 different methods that have been confirmed to show that they can be taken up in that manner. And each one has impacts on where it's going to go. I think the only thing that I was able to offer to him is that I have no direct evidence of this, but it appears to me that the exosomes will deliver their contents to the cell, and then most likely the lipid components, the membranous components, are then shuttled to the lysosome for degradation. And the only argument I have for this is that when we do our in vivo bioactivity experiments, we see extremely significant results in which in the heart, the scar is far reduced, and the radiation, the survival is incredibly increased. So we see the bioactive effects, which to me would indicate that they're not just being shuttled to the lysosome. But that is a natural progression of the uptake. So I think I mentioned to him, I'd love if he and his group would do that work, because they're fantastic. But somebody does need to on the uptake side of things, because it's a bit of a mess last I looked.
Okay, I think also, Marco, with your reepithelialization protocols, I mean, there is an impact on epithelial cells, at least, so, and also on T cells and others. So it means that maybe it's not immediately degraded by the cells.
No, you see, I think the functionality, a lot of people demonstrated clear functionality. And I was also struck by the instantaneous effect these vesicles have. So I did not discuss it now because it was more now on the challenges. But there is a difference between what I call ready-to-act cargo and cargo that needs more time to do something. Like the ready-to-act cargo are the proteins and the metabolites that are present. And indirect cargo is maybe the microRNAs that need to really modulate, and also the messenger RNAs, et cetera, but with these milk EVs, it's striking how fast they act. So that is also for me the point, and I did not have enough time about that, this spatio-temporal effect of the different cargos, that I think there is the secret of the vesicles, that they have both. They have ready-to-act cargo if you have an interaction and you can signal from the surface already if the vesicles fuse with the plasma membrane, which has also been described. Metabolites and proteins come directly available inside the soul. It has also been described that they can be endocytosed. You can have back fusion in the endosomal route. You can have lysosomal degradation. But if you then plot it to all the different cargo types, they also need to act at different parts in the cell. And a vesicle has it all. So I think that is also why people think the field is a mess, because there is not only one microRNA that you need to trace, because in the back of the microRNA are the metabolites, the proteins that signal from the surface, there are the coronas, and also the other colloidal things that can influence the uptake by the cells. So it is very difficult. But the good thing is that we do see function. We see effects.
Definitely. Okay, I think maybe last question or a couple of last questions. First one for Renk. EVs often relate to as having a homing capabilities, so they are home to the injured tissue, cell, organ. Can we have your insight about these capabilities? How does it happen? Or
well, there's a famous example in the hematology field that if you activate platelets, the platelets will expose receptors like P-selectin and the ligand for P-selectin is present on monocytes. And actually, this receptor P-selectin is sorted to extracellular vesicles from platelets. So the moment you get these platelet vesicles exposing P-selectin, they will almost immediately bind to monocytes and they activate tissue factor production, IL-6 production, etc. So there for sure are all sorts of fine-tuning mechanisms there to regulate where the vesicles are going. So it's like an envelope. There's really an address on it that it goes to a certain cell type. Yes.
Interesting. So maybe one last question for each and every one of you. Can you talk about the next steps the scientific community could address to advance the field? Maybe Rank, maybe we'll start with you and then we'll go through.
Well, I can. One of the things that has been lacking in the field for a long time, I think, is that we all have been measuring vesicles, the properties of vesicles, but results have been incomparable between laboratories. And at least now, we just completed an interlaboratory comparison study, a global interlaboratory comparison study, measuring concentration of vesicles with flow cytometry. And I think for the very first time, in a fully calibrated manner, we can show that we can measure all over the world the same concentration of vesicles in the same sample. And I think it's perhaps scientifically not so shocking, but I think also from a clinical perspective, it's mighty handy that you in the end measure all the same. I think that was something, a sort of anchor point that was missing. I think it's now there. And I hope soon that we get more of these tools that we really start to be able to compare data between each other. I still think that there are too many single center studies published with not reproducible results.
Thank you. Steve?
Yeah, I think
yeah,
I would build off of what Marka said, which I think was really well put. We have really good evidence that vesicles, exosomes have a dramatic biologic impact. And I think we still don't have a great handle on what the contents within them are that are doing that from my perspective, right? And when I think about like drug development, again, just as a clinician, like the critical step to take a concept all the way through the FDA and have a drug is you need to really be able to describe in detail from start to finish, not only that it has a positive effect on your patients, but how that is exerted. And so really like teasing that apart, I think is for me as a clinician, a critical next step.
Spencer?
Yeah, I think touching on that as well as what Marka mentioned, I think communication with the regulatory agencies is really, really important. And as of now, I mean, at least from my experience with the FDA in the United States, there just aren't codes and there aren't pathways at the moment because it's just slowly moving forward. So I think having more groups engaging with the regulatory agencies to define the pathways and get these things set in place so we understand the expectations, I think at least from the pharmaceutical side in the industry, would greatly accelerate the ability of these to move into the clinic.
Marka?
Yeah, then I come with a biological point. I think for me in the future, it is really the interaction between the vesicles and all the colloidal structures. So I call it, now we have been working on biogenesis, the origin from the cell, but there is a big gap in the journey. What happens once the vesicle is released by the cell? And what is the journey of the vesicle in the tissue and in the biofluid influencing the targeting and the uptake and the delivery of the specific cargo? So I would like to go on the journey and to the target cell, to the zipper, what's going on there.
Thank you very much for all of you. Unfortunately, we must bring this discussion to a close. So I would like to thank again to our esteemed speakers for their informative and inspiring and open contribution to the audience and to the field in general. And before we finish, I will give back the floor to Jennifer Smilowitz for closing remarks.
Okay, can you see my screen?
Yes.
Awesome. Okay. Hi, everyone. I'm back. Wow, what a riveting set of talks and discussions on the challenges, but also the potential of milk EVs and microRNAs for their clinical applications, which is what we are very interested in. Please note the recording of today's webinar will be available on the Milk Genomics website shown here around June 1st. So you will receive an email from me around then, and look out for the recording. So, the topic for each year's Ross Roll Up Your Sleeves workshop is partially selected through a voting system from our previous year's workshop attendees. That's all of you. But the speakers for the workshop are selected by the IMGC Roll Up Your Sleeves workshop committee. And this year's workshop committee included members from the Scientific Advisory Committee for the IMGC, and that includes Professor Nuri Argov, our fantastic moderator, Professor Casper Hedinga from Wageningen University and Research, and Dr. Layla Strickland, the CEO of BioMilk. And I would like to give a big round of applause and thanks to this committee for their many hours spending researching the topic and finding our amazing speakers. So thank you very much. So as you know, we are a nonprofit organization, and in order to function and pay for all of our platforms, we rely 100% on sponsorship support. And so I'd love to give big thanks to our 2023 elite sponsors, whose generosity has been instrumental in supporting our advancements in the fields of lactation and milk science. And this year's elite sponsors include Arla, ByHeart, California Dairy Research Foundation, which also co-manages the IMGC, and finally the National Dairy Council. And if you're interested in sponsoring our activities, please contact me anytime. And then finally, scientific advancements could not be made without the brilliant and passionate innovators and scientists like all of you. So we rely on our base to help us advance the field. And our success is dependent on your insight, your input, and engagement. And there are 5 ways that you can be involved in this organization. So first, if you aren't already, please subscribe to Splash Milk Science Update, and that's our bi-monthly publication on milk science for human health. And also please share the articles through your social media network. This is so important. Oh, sorry. Next, please complete the 5-minute exit survey. And this is so important because your vote can help us identify next year's topic for the workshop. And another way you can engage is to join our various working groups on co-authoring peer-reviewed review papers. So if you're interested in being a co-author on any of our working groups, just list your name on the exit survey. Now, I hope to finally see you all either in person or virtually at our upcoming 20th annual IMGC Symposium, which is hosted at University College Cork in Cork, Ireland on September 6th to the 8th this year. And so thank you all for being here, for participating, and we'll see you soon. Bye.